Institute of Microbiology, University of Agriculture, Faisalabad, Pakistan. So this is a pioneer institute in Pakistan which aims to carry out the teaching and research activities in the field of virology, immunology, mycology, vaccinology, and biotechnology. So this is a brief introduction about myself. I'm currently working as a lecturer and a PhD scholar at Institute of Microbiology, Faculty of Veterinary Science, University of Agriculture, Faisalabad, Pakistan. My major fields of specialization are the virology, vaccinology, and molecular biology. So I'm working in the cultivation and characterization of infectious bursal disease virus, and my PhD it is based upon the immunodiagnostic and vaccine development in the field of you can say the virology. And two major viruses I am concerned about. These are the infectious bronchitis virus and infectious bursal disease virus. These are the current research projects that are the development of indirect ELISA using recombinant VP2 protein of infectious bursal disease virus and microbial assessment of potable safe water in the major districts of Punjab, Pakistan, and the development of monoclonal antibodies. It is antibody-based technology for the rapid and differential diagnosis of infectious bursal disease and Newcastle disease virus. As far as this study is concerned, so the infectious bursal disease virus, it is a disease of poultry birds that have major impact as an atrophy and necrosis of bursa fabricius. First, this disease was discovered in USA in 1962. So the basic structure of the virus, this is non-enveloped bisegmented double-stranded RNA virus that have 32 capsomer having a diameter of 60 to 70 nanometer. If we study the structure of this virus, so this virus is composed of five virion proteins. These are known as the VP1 protein, VP2 protein, VP3 protein, VP4, and definitely the VP5 proteins. VP1, it's basically the RNA-dependent RNA polymerase of the virus. And the VP2 protein, this is the major protein that behave as an antigenic protein. So this forms the major structure of the virus that form a trimeric subunit with the VP3 proteins. VP4, it's a non-structural protein, and this is a minor virion protein that helps in the processing of VP2 protein, VP3, and VP4 itself. VP5, it's a virion protein 5 that is involved as in the dissemination and release of the virus, but its structure or function is still unknown. This is the two segment of the virus that contains segment 1 and segment 2, which is A and B segment. That contains mostly the three open reading frame. Open reading frame 1 that composed of VP5 protein, and open reading frame 2 that encodes pre-VP2 protein, VP4 and VP3. While segment B, it contains open reading frame 3 that comprises of VP1 protein. After the infection of this virus inside the body of the bird, this virus starts replicating in the gut-associated lymphoid tissues, and via the blood, it affects and destroys the beta cells. That definitely leads to the immunocompression or immunosuppression that could be lethal, definitely for birds. This virus was first time observed 40 years ago, and till now this is causing the continuous effects to the poultry industry. So the only way to avoid this type of viral disease is the vaccination. But for vaccination or for the effective use of those kind of vaccines, we have to develop certain techniques just to know about the actual antibody titer present inside the body of the birds. So there is a most reliable method known as the ELISA method. This is enzyme-linked immunosorbent assay, which is based upon antigen and antibody reaction that would be indicated by the change of color. So its application is highly sensitive in the detection of this disease-related antibodies, and ultimately it can help out in the quantification of the virus. The aims of this study is to optimize the test conditions for the indirect ELISA, the quantitative determination of infectious bursal disease antibody titer in the serum, and the evaluation of the vaccine performance. So in this study, we mainly focused on the antigenicity or the major VP2 protein to detect the anti-infectious bursal disease virus antibodies by the help of this developed ELISA, which serve as a chemically defined, safe, and non-infectious serodiagnostic method. This material method composed of sample collection that includes the collection of bursa samples from the infected flocks, histopathology for the identification of you can say those type of atrophy of the bursa or necrosis of the bursa, virus RNA extraction, cDNA senses, PCR amplification, then the gel electrophoresis to visualize and confirm the VP2 protein or VP2 region of the infectious bursal disease virus. And this product will be again amplified by gel purification method, and that includes the isolation of plasmid from the E. coli bacteria by alkaline lysis method and restriction digestion of that plasmid and the DNA. The ligation of this DNA and PET28A, which is a vector, preparation of competent cells, and transformation of this ligated product into that PET28A. The cloning PCR that indicates the virus after cloning, sequence and phylogenetic analysis, recombinant protein expression on STS, and purification through the nickel affinity column, and that purified antigenic protein of VP2, it will be coated on indirect ELISA plates that will be evaluated against the vaccinated birds. These are the primer sequences that are designed by utilizing the laser gene softwares. This is a forward primer sequence and the reverse primer sequence for the cloning purposes. Then comes the amplification of this product by PCR technology. The annealing temperature, it is about 55 degrees centigrade for 45 seconds, and extension at 72 degrees centigrade for 45 seconds, and that includes the final extension at 72 degrees centigrade for 10 minutes. Then comes the restriction and digestion of plasmid and the virus itself. So this is performed by cutting the band of a gene of interest for the cloning while leaving rest of the region. These restriction sites are added in the primers that amplify the virus through PCR, while those sites are naturally present inside the plasmid, which is PET28A. The restriction sites that are used in this study are BAMH1 and EcoR1. These sites are compatible to the cut genes that are present in the primer region also. Using T4 DNA ligase enzyme, the viral DNA was inserted or ligated inside the plasmid DNA. The recombinant plasmid, it contains the viral DNA. It is transferred into the expression vectors, and the expression vectors are the two strains of the E. coli bacteria. These are BL21 and DH5 alpha. So here are different results. The first picture indicates about the postmortem examination of the bursa that indicates the inflammation of the bursa in the poultry birds. And the second picture indicates about the necrosis and eosinophilic infiltration of the inflammatory cells inside the bursa after histopathology. These are the different amplification pictures, the amplification product by consensus primers, and then confirming these PCR product by the help of partial VP2 primers. And the third one indicates the 5500 base pair band size of PET28A plasmid. These two pictures indicates the recombinant plasmid that are expressed in BL21 E. coli strain, and this media contains the kanamycin as a resistance agent. So this is the clone PCR picture that indicates the clone. After cloning PCR, this picture represents about the amplified product of partial VP2 region, which amplified and gives the band of 699 base pair. After confirmation of and sequence analysis of this VP2 region, this VP2 region have some similarities after blasting this region to NCBI, and the phylogenetic analysis indicates this clone of VP2. It is related to very virulent strains of India, Pakistan, and Bangladesh. So this is IPTG induced E. coli and protein expression after STS. And after confirmation through STS, we have purified this protein.
I'm sorry, you've got 1 minute. Please conclude.
Okay. After confirmation through nickel affinity column, then we have coated this on a ELISA plate, and after ELISA, we have optimized the different dilutions. The 25x dilution, this will be taken to coat on this ELISA plate, and this can be tested after vaccination of the bird with the Bursaplex vaccine. The mean values of this titer indicates the up to day 14, there will be a maximum increase in the antibody titer that was detected by this developed indirect ELISA kit. This is the conclusion. That includes the optimized conditions for ELISA. It includes 30 microgram per ml of the proteins that is coated and 100-fold dilutions of the serum. So this type of developed ELISA, it gives the antibody titer that indicates the antibody in the serum. It is increasing from day 3 to day 7, which is maximum, and then start decreasing after day 7, after day 14. Sorry. The approximate cost for this type of ELISA, it is about 10,000 Pakistani rupees, while the actual cost of commercially available kit in Pakistan, this is about 40,000. So the work is still going on to determine the efficacy and sensitivity of this developed ELISA as compared to other commercially available kits. So these are the two research papers published already in 2 impact factor journals. Thank you.
Thank you for your presentation. Yes, if you have a question for Dr. Sanallah, you can ask him. If not, we will move to Dr. Habib Jurdem.
Yes. Yes, I am.
Yes, you can share your screen and start.
Yes. No, it's here.
Yes, it's visible. Can you make it full screen first?
Full screen? Yes.
Full
Now we
screen.
can. Yes.
It's okay. You can start. You've got 10 minutes. Yeah.
Yeah.
Okay, great.
I am Habib Jurdem from Laboratory of Fundamental and Applied Mathematics of University Oran, Algeria. My presentation titled Solvability of Nonlinear Singular Fourth Order Two-Point Boundary Valley Problem. We divide this presentation on 3 sections. In the first one, we introduce our problem. Then we give some preliminaries that we need it in the proof of our results. Bending or deformation of elastic beams and other important applications in mechanics, engineering, and physical science can be modeled as a problem of fourth order ordinary differential equation. Many authors have studied the beam equation under various boundary conditions and by different approaches. Now we define, the aim of this work is to study the existence of positive solution for the singular fourth order two-point boundary valley problem noted 1, subject to the boundary condition in 2, where lambda is a positive parameter and A is a continuous function, F a non-negative real valued function. Under various assumptions on A and F, we establish intervals of the parameter lambda, which yields the existence of at least one positive solution. Our main tool is the Krasnolevsky fixed point theorem of cone expansion compression cone. Consider the Banach space of all continuous function unloaded with the maximum norm. In the first lemma, the linear problem 3 can be reformulated by the integral equation ut equal integral from 0 to 1 gts ys ds, where g is the green function, which is in 4. In lemma 2.3, we approximate the green function g for all ts in intervals 0,1 times 0,1. For theta between 0 and 1, and y is a non-negative continuous function, the unique solution ut of the linear problem 3, 4 is non-negative and satisfying this inequality. Now we define the cone K and the operator T lambda, which is in 6. A solution of the nonlinear problem 1, 2 announced in the introduction is a fixed point of the operator T lambda. Then we use the following assumption. In the first one, H1, we suppose that the integral 0 to 1 of G1 SAS is infinite and it is positive. In H2, we suppose that f is continuous and the function a is not vanishing. For that, there exists T0 in 0, 1, which such that a T0 is greater than 0. In lemma 2.5, we show that in lemma 2.5, we show that T lambda K is T lambda associate each function in K with a function in K. Then in lemma 2.6, if H1, H2 hold, the operator T lambda is completely continuous. In this demonstration, we show that the condition of Ascoli theorem, Ascoli-Arzela theorem are satisfied. Now we present our main result, called the Krasnolevsky fixed point theorem of cone expansion and compression type. If assume omega 1, omega 2 are two subsets of A with element 0 in omega 1, and the closure of omega 1 is included in omega 2. T is a completely continuous operator. If it is satisfying one of this condition, A to be said compression, B to be said expansion, then T has a fixed point in this subset. In the remainder, we use the following notation: F0 said be said if F0 equal 0 and Ft equal Ft, we said that F is superlinear. If F0 equal Ft and Ft equal 0, we said F is sublinear. Now we present our result. If H1, H2 hold, and lambda is a positive, real positive number, the nonlinear boundary value problem 1, 2 has at least one solution in the case E or 2E.
You got 1 minute left.
The proof if this theorem is based on Krasnodębski fixed point theorem. In theorem 3.2, with the same hypothesis H1, H2, and this double equality, and lambda is in this interval, the nonlinear problem 1, 2 has at least one positive solution. To complete the proof of theorem 3.2, we use the corollaries 3.3, 3.4. The last theorem 3.5, under the same hypothesis and this double equality with lambda in this interval, the nonlinear problem 1, 2 has at least one positive solution. To complete the proof of this theorem, we use these corollaries. In the final, we have this reference. Thank you for your attention.
Thank you for your presentation. Do you have any question for Dr. Habib? You can stop sharing your screen. Thank you. So we move on to the next presentation. I don't think that Wafia Wafa is here and her group. So we move on to Yasham Abdelmoujid. Are you here? Yes, Yes, I see you. If you're ready, please share your screen. It seems that he's not ready. Dr. Yasham Abdelmoujid, we can't hear you. Okay, we move on and we get back to you when you're ready. We move on to the next presentation. Dr. Sehrish Firyal, are you here? No, I don't see her too. So the next will be Dr. Bedani Hamid. Are you there? Yes. You need to unmute yourself. You need to turn on your microphone.
Yes, I am here, yes.
Yes, we see you. Can you make your camera properly and then start sharing your screen?
Yes.
By the way, Dr. Stefan, do you have presentation here? I don't see your name on the list. Dr. Stefan. All right, doesn't have.
Hello, everybody. I'm Dr. Bedani Hamid, professor at the Algeria University. I present to you this work entitled Fractional Domain. We consider the fractional differential equation, next equation, where
Well, it's so noisy over there. We can't hear.
To the capital fractional derivative of order alpha between 0 and 1. And Z is complex number as Z, BZ, B on T fiction. We will find conditions on BZ as Z which will guarantee that every solution F different 0 of the equation we will have infinite order. We will address the following points: introduction, preliminaries, existence, and results. The author study various types of fractional differential equations in complex domain, such as the Cauchy equation, the division equation, and telegraph equation. Transform is a significant technique to solve mathematical problems. Manifold transforms for solving various problems appear in open literature such as Y formation de Laplace transform, the Fourier transform, the integral transform, the local fractional integral transforms, and the fractional complex transform. In this section, we introduce some notations and definitions for a fractional operator, derivative and integral in the complex Z plane as follows. Definition 1, the fractional derivative of order alpha is defined for
Dr. Bidani, you're gone. It seems that you've got an connection problem. Hello? Yes. His screen is frozen. I think. We've got to wait because he's not gonna hear us. Yes, he's disconnected. So yeah, who's the next in the list? If I called Wafa, I think she joined after. If you're ready, please share your screen. Do you hear me, Dr. Wafa? It seems that she doesn't. Okay, then Dr. Gesham, are you there? If you're ready.
Hello.
Yes.
I am sorry, problem technique connection.
Yes, you've got a connection problem. If your connection is not that good, you can keep going orally without sharing the screen. All right, sharing again. Dr. Wafa, let's wait and you'll be the next.
The definition 1, the fractional derivative of order alpha is defined for fraction fz by relation 1.
Yes. Yes.
Yes, yes. Definition 2, the fractional integral of order alpha is defined for a function fz by relation 2. Applying the Capito method, we know the Capito derivative by the formula 3, where alpha between n minus 1 and n belong in n star. In this work, we assume that the rider is familiar with the fundamental result and the standard notation of the Livanina value distribution theory of meromorphic function. See the references. Let rho f denote the order of an entire function f is relation next, where TRF is the Livanina characteristic function of f, where MRF equal max module fz equal f. For example, the function fz equal expose z square satisfy rho f equal 2. Relation 4. Lemma 3, let f be a transcendental entier function of finite order rho, let omega defined as denote a finite set of distinct pair of integers that satisfy relation, and let epsilon greater than 0 be a given constant, then the following three statements hold. There exists a set A1 connected interval 0 to P that has linear measure 0, so that if T0 belong in then there is a constant R0 that for all z satisfying argument z equal phi 0 and modif z is greater than R0 and for all kg belong in gamma we have relation phi. Consider the fractional differential equation 6, where the alpha z and the beta z are the capital fractional derivative of orders alpha and beta belong in n-1 and n, and alpha different beta and belong in n star, and z is complex number as z dz be entier function. Theorem 4. Let be entier function such that for real constants lambda mu theta 1 theta 2 where lambda positive mu positive and theta 1 less than theta 2 we have relation 7 and relations next as z to infinity in argument z between theta 1 theta 2 then every solution f different 0 of 6 has infinite order. This is the reference. Thank you very much for our attention.
Thank you for your presentation. And if you have any question, you can ask. If not, we will move to one of you, Dr. Gasham or Dr. Wafa, who is ready. Dr. Gasham, it seems that you have a problem, so you have to leave room and then try to solve it with my friend at the reception, because we can't hear you. All right, Dr. Wafa, can you start?
وعافية وفا راهي تهضر معاك بعد ذلك. وعافية وفا.
Yes, I am here.
Yes, please. You can share your screen.
I am here. Hear me, please.
Yes, it's coming. Your screen is coming. We don't see it yet.
Can you hear me, please?
We can hear you, but we can't see your screen.
Yeah, I tried.
Maybe you need to stop first your screen and then start again. All right, now we see it. It's visible. Yes, you can start. Hello?
Can I start?
Yes, please. Yes. And you've got 2 presentations. She doesn't hear us, it seems. All right. It seems that she's also, yeah. Do you have connection problem? All right. So who is ready? Dr. Wafa, do you hear me? it seems that you've got a connection.
Yes.
So you can
Do
make,
you see my slides?
no, we can't. You can just do it orally without showing your screen. Okay, now we see it. You've got 10 minutes.
Yes,
Please
I
start.
can. Yeah. Yeah.
All right, if Dr. Hadil is ready. Are you in this session, Dr. Hadil? I can't see your name on the list.
هديل إذا مستعد بابا.
It seems that she doesn't hear us. And Dr. Abdul Mujib, do you hear me?
Yes, yeah, I am hearing you. You hear me?
Yeah, you can start your presentation, please.
Okay.
You'll be the
So I
next
can
Dr.
start.
Hadil. It's coming. Yes, we see it now. You've got 10 minutes.
Okay. So firstly, thank you for your acceptation to present my work in the conference. My presentation is about the effect of demethylation agent, which is 5-azadoxystrobin, which can reduce the viability of colon cancer cells. So in my presentation, I will start firstly by presenting the characteristic of cancer cells. Generally, cancer cells are characterized by activation of oncogenes and inactivation of tumor suppressor genes. This inactivation can be caused generally by hypermethylation of promoters. So the hypermethylation of promoters can be modulated by epigenetic modulation treatment. So our
Again, a connection problem.
So you hear me, please?
Yes, you can keep going without sharing your screen. It seems that your connection is not that good.
Okay, that's good. Okay, so the object of our work is to study the effect of demethylation treatment on colon cancer cells, and to study also the difference of this effect on metastatic and non-metastatic cells. So two steps different of cancer. In our so we from colon cancer, of course, which are CACO2 and LAVO obtained from ATCC USA. The CACO is cultivated in DMEM and LAVO in EMEM. Also, we are using an epigenetic modifier, which is 5-azadoxystrobin, which is an methylation inhibitor. So the methylation inhibitor 5-azadoxystrobin is considered as cytosine analog, so it can stop the methylation by adding on cytosine. So our methods, so we can resume the methods we are used. So the cell culture, we culture both cell lines, CACO, which is not metastatic, and LAVO, which is metastatic. and reaching the confluence. After reaching the confluence, we use the treatment of the demethylation agent 5-azadoxidine in 0.2 micromole for 72 hours. And we have made the viability test by using the Risazine basic test. Here results about the viability that we find that the demethylation agent 5-azadoxidine can reduce the viability and we are used the H2O2 as positive control, and we found that metastatic cells are more sensitive to epigenetic treatment than non-metastatic CACO. Regarding the results, which we find that there is no different significance on the viability of cancer cells with this dose of 0.2 micromole, but we find that metastatic cells of colon cancers are more sensitive to epigenetic therapy than non-metastatic cells. Studies that prove that the methylation agent can reduce cancer cells viability, especially after treatment with methylation inhibitors in different stages of cancers. The viability also find that we have made another experiment about the DNA damage and DNA repair. We found that the viability of cancer cells, colon cancer cells, by demethylation agent may be result by DNA damage, the DNA damage, or also inhibition of DNA repair also. Also, we suggest that the combination of other epigenetic modification may be by
Again, a connection problem. All right. Yes, he's disconnected. All right. Who is ready? Dr. Hadid or Dr. Wafa? Who is ready between both of you? Please turn on your microphone and start.
Hello.
Yes, we hear you.
I'm ready.
Who's this?
Buafia Wafa.
All right. Okay, please share your screen. It seems that we have got really serious connection problem. All right, please don't share your screen. All right, she's gone again. Dr. Yasham, are you back? Okay, Dr. Hadid is faster than all of you. All right, okay.
My study is simultaneous approximation of a modified family of the ZAS Markov beta type operators. Approximation and some other fundamental properties of linear and positive operators are potentially useful in many different research areas in the mathematical, physical, and engineering science. For f in C of 0 infinity, the Zaz operator defined as follows, where Qn Kux defined as follows. This family studies in by Markan several modification papers of Zaz-Markan type operators were published. Some others extend the classical gamma and beta functions. For example, Chaudhry and Zubair introduced the following extension of the gamma function. And Chaudhry and others presented the following extension of Euler's beta function. Many extensions and the generalization of gamma and beta functions are studied. For this, we refer to many references. Recently, Surfista and others introduced and studied an extended family of the Zaz-Markan beta type operators in ordinary approximation, which defined as the follows. This paper studies the fundamental convergence theorem in simultaneous approximation for the operators which studied by Surfista Sn star of f will x in the space C alpha of 0 infinity, which is defined above. Then the approximation order via Wronowski type asymptotic formula shows that big O n of power -1. Finally, estimating the error occurred by this approximation is given in terms of modulus of continuity. Primaries. In this part, we give some definitions and the theorems which we need in our study. In Lemma 2.1, this is the results find from Surfista follows for x equal to for x in 0 infinity, the following condition hold. In the reference 17. In Lemma 2.2, for m we obtain Sn star of t of Tm will x equal to n minus m factorial over n factorial nx m plus m of m equal addition 1 nx m minus 1 plus big O n minus 2. Definition 2.1 for m in n naught, the mth order moment mu n m of x for the operator Sn star of f will x defined as. Lemma 2.2 for the function mu n m of x, we have mu n0 equal to 1, mu n1 of x equal to 2 over n, when mu n2 follows as. In Lemma 2.3, there exist polynomials qi jr of x independent of n and k such that xr dr qn k of x equal to summation ni k minus nx j qi j rx qn k of x. Main results. In this part, we show the derivative dr over dxr of Sn star of f will x is an approximation processes for fr of x will x in 0 infinity. In theorem 3.1, for r in n will f in c alpha of 0 infinity for some alpha less than 0, and FR exists at a point X in 0 infinity, then we get the follows. Theorem 3.2, if F in C alpha of 0 infinity for some alpha leg of 0, if FR plus 2 exists at a point X in 0 infinity, then we get the follows.
Please move to the conclusion.
The order of the error of order of infinity of approximation we get by the theorem 3.2. Let F equal in C alpha of 0 infinity, we get the follows.
Can you move to the
And this is
conclusion,
my
please?
references.
Conclude. All right, thank you. Right.
انتقلي للمناقشات. أكو مناقشة؟
Okay, thank you. If you have any question, please go ahead. And yeah, we still have the connection problem with Dr. Wafa. All right, we see your screen. You can start. You've got 10 minutes. Oh, you've got 2 presentations, right?
You can hear me, please?
Yes, we do. Please start.
My present work. Yes. It's my work is by bisulfonyl flavonoid contents and antibacterial activity of the extract of Ephedra altissima. The human multitude of that can invade its tissues under certain conditions, causing serious infection disease. The therapy of bacterial infection is mainly based on the use of antibiotics, which selectively inhibit certain metabolic pathways of bacteria without exerting toxic effects on the human body. In fact, the inappropriate use of this agent can generate multi-resistant strains. For this reason, several research studies are focused on the discovery of new herbal medicines that can effectively combat the phenomenon of multi-resistance. So the genus Ephedra belonging to the Ephedraceae family, including approximately 68 species, is characterized by perennial plants with shrubs. Ephedra altissima is dioecious, climbing, much brown. It is a high climbing giant fire species used in folk medicines for the treatment of various diseases such as vascular hypertension and respiratory disease. The objective of the present work is to describe a preliminary phytochemical investigation, the estimation of total phenolic and flavonoid contents, and the evaluation of the antibacterial activity of the ambitanol extract. Materials and methods. So for the extraction procedure, the aerial part of the species Ephedra is macerated twice in mixture solvent ethanol water for 3 days. Filtration, the filter obtained extraction using 3 solvents with increasing polarity. Then the fraction was evaporated and acetate and ambitanoic extract. The presence of different classes of secondary metabolites contained in the ambitanol extract were quantitatively tested using the methods described by Farnsworth. The estimation of total phenolic and flavonoids contents were carried out using the Foloncino-Calto and trichloroaluminum methods. And the antibacterial activity was carried out against 7 bacterial strains, including 3 gram-positive bacteria, Staphylococcus aureus, Staphylococcus pneumonia, and Streptococcus mittens, and 4 gram-negative bacteria, 3 of them Escherichia coli and 1 Pseudomonas aeruginosa, using agar-disc diffusion assay, and the minimum inhibitory concentration were determined by microdeletion method. From the result and discussion, the phytochemical screening of the ambitanol extract revealed the presence of tannin, flavonoids, coumarin, and saponin, while the alkaloid, quinones, and anthraquinone absent in Ephedra altissima plants. The richness of the extract with secondary biological activities. For the total phenolic and flavonoid content showed value of 125 microgram equivalent gallic acid per milligram and 16 microgram quercetin equivalent milligram of plant extract respectively. Contents of phenols and flavonoids. Several studies carried out on total phenolic and flavonoid content from species of the genus Ephedra showed variation in their level. We have Ephedra major, Ephedra disticha, Monosperma fragilis, Phoeminea, Alata foliata, vulgaris, and Ephedra sinica, indicated the lowest contents of phenols and flavonoids compared to the current study. When Ephedra sarcocarpa, Strobiliacea, Pachyclada, Laristanica showed or displayed a higher content of phenols than Ephedra altissima. So the observed variation is in the results of total phenolic and flavonoids contents might depend on several factors such as soil characteristic, period and area of harvest, plant storage condition, and analytical quantification methods. For the antibacterial activity, the ambitanol extract had antibacterial activity against all the gram-negative strains and only against the gram-positive bacteria Streptococcus mittens. The antibacterial strain Pseudomonas aeruginosa was the most sensitive, with a value of MIC at 3 microgram per milliliter. So we have this table present the antibacterial of the crud extract altissima against gram-negative bacterial strain. It have antibacterial activity against all the strain used in this study. When the table 2 present the gram-positive bacterial strain, it shows only activity against Streptococcus mittens with MIC 25 microgram per milliliter. The ambitanol extract exhibited antibacterial activity against the most tested bacteria. This activity could be explained by the presence of different phytoconstituents such as flavonoids are known for their antibacterial properties. The phenolic compound can act at 2 different levels in the cell membrane and the cell wall of microorganisms. These compounds can also interact with the membrane protein of the bacteria through hydrogen bond, causing changes in the membrane permeability and cell destruction, penetrating bacterial cells and coagulating the cell contents. Furthermore, flavonoids are well known for their role in retarding the growth of microorganisms by inhibiting their nucleic acid synthesis, the function of cytoplasmic membrane, and energy metabolism. When the lack of the antibacterial activity of the ambitanol extract against some bacterial of the strain, positive strain can be explained by the choice of the methods, condition, and the use of tools to perform the test. So the method from Wells and agar is more suitable for studying the antibacterial activity of aqueous and organic extract than the diffusion method in agar medium. And in this study, we use the method of agar medium. However, it is possible that the addition of dimethyl sulfoxide to plant extract reduce its antibacterial activity. So this is the name of the study who have also found antibacterial activities to the genus Ephedra altissima. So, as a conclude, we can conclude the phytochemical screening of the ambitanolic extract revealed the presence of several classes of secondary metabolites. Furthermore, the extract showed an antibacterial effect against at least 3 strains. Altissima extract will be used as an important source of natural bioactive molecule with antibacterial properties.
Thank you for your presentation. You can stop sharing your screen. Dr. Hesham, you can just move to the conclusion because it seems that you've got a serious connection problem. But first, Dr. Waf needs to stop sharing her screen.
Yeah.
Can you stop sharing this? This is your screen, right, Dr. Wafa?
Now I stop the share.
All right, thank you. Yeah, it's gone. All right, Dr. Hesham. Hello? Dr. Malki, you have joined. If you're ready, please share your screen. Dr. Sanaullah, you can leave if your meeting time has come.
Thank you so much.
You're welcome. Dr. Hesham or Dr. El Malki, any of you is here? You're the last ones. All right, it seems that they're not here. Or maybe they have got a connection problem. So yes, thank you all for participating in our conference. Hope to see you again. Take good care. Bye.